Welcome to Lecture 17, where we will go over the processes of osmoregulation and the excretory system. As stated previously, blood will be brought to the kidney by the renal artery and will be removed from the kidney by the renal vein. The outer renal cortex surrounds the inner medulla. Both of these areas, the cortex and medulla, contain nephron tubules and capillaries for filtration and resorption. Cortical nephrons, please refer to the diagram to the right, are much shorter and more abundant in the kidney than the juxtamedullary nephrons. Note the difference between the two nephrons. The juxtamedullary nephrons will have the tubule that penetrates deep into the renal medulla, whereas the cortical nephrons have much shorter tubules. Urine is considered to be hyperosmotic to the body fluids. This helps maximize water conservation, which is considered to be a key adaptation for terrestrial living. We're now going to take a look at the structure and function of the nephron. Remember that blood is being brought to the kidney through the renal artery. This will branch into the afferent arteriole, which will then lead to the capillary network in the Bowman's capsule. Also keep in mind that the arterial flow has a higher blood pressure. This blood pressure will force blood from the afferent arteriole into the capillaries and the glomerulus. Fluid will then be forced out of the capillaries, and this fluid will then collect within the Bowman's capsule. The filtrate will then be processed as it travels through the tubules, specifically the proximal tubule, loop of Henle, distal tubule, which will then enter into the collecting duct. Keep in mind that blood vessels, such as the peritubular capillaries, as well as the vasa recta, help to surround the nephron to resorb different types of solutes and molecules. The filtrate will then empty into the renal pelvis. This will then drain into the bladder through the ureter. We're now going to take a look at the steps involved in urine formation. We begin with looking at the proximal tubule. The proximal tubule is a site of the reabsorption of ions, water, as well as nutrients. It also involves the secretion of hydrogen ions as well as ammonia. This helps to maintain the overall pH balance of the blood and tissues. It also helps to remove any drugs or toxins from the liver. In the proximal tubule, the filtrate will become concentrated. In addition, small amounts of ammonia, NH4, will be produced. Take a look at the diagram to the right to understand the movement of salts and water through active and passive transport. Passive transport involves the reabsorption of carbonate, water, as well as potassium. Ammonia will be passively crossing the transport epithelium to enter into the lumen of the tubule to be eliminated. Hydrogen ions will also be pumped into the lumen, and sodium chloride as well as other nutrients will actively be transported out of the lumen into the surrounding tissue. The second step in urine formation occurs in the descending loop of Henle. In this region, water will be reabsorbed through aquaporins in the transport epithelium that lines the tubules. This is driven by the hyperosmotic conditions of the interstitial fluid surrounding the tubule that increases as you move towards the inner medulla. Salts and other solutes are conserved, and the filtrate becomes further concentrated as it moves down the loop. As the filtrate moves to the ascending loop of Henle, sodium chloride will be reabsorbed as it leaves the tubules. First, this transport occurs passively, but then it occurs actively as it moves into the cortex. This section of the tubule has no aquaporins, which means that it is impermeable to water. As a result, the filtrate becomes more dilute as it moves through the ascending loop. The filtrate then moves into the distal tubule. In the distal tubule, sodium chloride and bicarbonate will be resorbed into the surrounding tissue, mostly through active transport. Potassium and hydrogen ions will be actively pumped across the transport epithelium into the lumen of the tubule. As a result, water will then leave through passive transport, or water will be resorbed. This process in this area is highly important in regulating the concentrations of both potassium and sodium chloride in the body tissues. Because you have the transport of hydrogen ions, this will also be helpful in controlling the overall pH of the surrounding tissues and blood. The last step in urine formation occurs as the filtrate passes through the collecting duct. Urine concentration is highly regulated by hormones as the filtrate passes over the transport epithelium. This area can respond to the different needs of the body. If there is a need to conserve water, this can be done by increasing the permeability of the tubules to water molecules while restricting the passage of salts and urea. If there is excess water in the body, this excess water can be eliminated by absorbing salts and restricting the movement of water molecules. The same hormones that are involved in regulating blood pressure, blood volume, and osmolarity also play a role in regulating the concentration of the urine produced. Please take a look at this diagram to understand the overall steps in urine formation that were described in the previous slides. Note where active transport and passive transport occurs and the movement of important substances such as water, bicarbonate, hydrogen, potassium, ammonia, as well as sodium chloride. The structure and function of the kidney shows a high adaptation for terrestrial living. Mammalian kidneys can produce highly concentrated or hyperosmotic urine. This is very important in the conservation of water in terrestrial environments. However, the active transport of solutes, primarily sodium chloride, requires a lot of energy, which means that this is an energetically costly process for the organism. We're going to take a look at the osmolarity of the filtrate and surrounding interstitial tissue in the kidney. As the filtrate leaves the Bowman's capsule, it will have the same osmolarity as blood, which is about 300 milliosmoles per liter. As water leaves the tubule, the osmolarity of the filtrate will increase. This is what occurs as the filtrate moves down the loop of Henle. As the filtrate then moves back up the ascending arm, sodium chloride is resorbed. This will cause the osmolarity of the filtrate to have an overall decrease as it moves up the ascending arm. As the filtrate then moves down the collecting tubule, water is resorbed and the osmolarity will increase again. This leads to patterns of increasing and decreasing osmolarity, which will help in the conservation of water. This diagram reflects the movement of the filtrate as it passes through the nephron, as well as the osmolarity of the filtrate with respect to the surrounding tissue. Sodium chloride and urea are important in contributing and regulating the osmolarity of the interstitial fluid. This is then used to increase the kidney's ability to conserve water. Note that as the filtrate leaves the Bowman's capsule, it has the same osmolarity of blood of about 300. As it then moves down the descending loop of Henle, note the increasing value of the osmolarity. Also note that as the filtrate then moves back up the ascending arm, we have a decreasing osmolarity due to the passive and active transport of sodium chloride out of the lumen of the tubule and into the surrounding tissue. Note that at the distal tubule we have a very low osmolarity value. However, as the filtrate then passes down the collecting duct, we see an increasing osmolarity value. This is mainly driven by the transport of water or reabsorption of water into the surrounding tissue. We're now going to take a look at some adaptations that different animals have. Mammals that need to conserve water, like the desert mouse, will generally have longer loops of Henle, or more juxtamedullary nephrons. This allows them to produce very small amounts of hyperosmotic or highly concentrated urine. Because the filtrate needs to pass through a longer loop of Henle, this allows for the maximum reabsorption of water, thus maximizing the conservation of water for the animal. However, mammals that live or spend time in aquatic environments have the opposite. Mammals like the beaver will have more cortical nephrons. This is because they don't have as much of a need to conserve water. By having more cortical nephrons, they can produce a less concentrated or hypoosmotic urine. The vampire bat has unique adaptations that allow them to metabolize and use a blood meal. Keep in mind that a blood meal is also costly to not only metabolize, but to also house within the stomach. Heavier animals will need to use more energy to fly. As a result, the vampire bat will produce large quantities of diluted urine very quickly while it is feeding. This will help to metabolize the blood to ensure that it is light enough to fly when feeding is done. However, when they are roosting, they can produce very small amounts of highly concentrated urine. This allows them to stay light when active and also to eliminate urea waste products from the blood meals while conserving water. Birds that live in highly dehydrating environments like the albatross also have juxtamedullary nephrons. Note that the presence of juxtamedullary nephrons are considered to be unique in mammals, except they are present in some aves as seen in the albatross. However, these juxtamedullary nephrons do not extend far into the medulla, as we see in mammals, and cannot concentrate the urine fully. As a result, they produce uric acid in order to help them conserve water. Also keep in mind that we also examined in previous slides the presence of the salt glands in the nasal cavity that also help to conserve water in a dehydrating environment such as the marine environment. Other reptiles have cortical nephrons that produce iso or hypoosmotic urine. However, the epithelium of the cloaca can help resorb and conserve water from the feces and urine mix before it leaves the body. Reptiles also have the ability to excrete uric acid, once again increasing their conservation of water in different types of environments. If you recall, freshwater fish need to maintain hyperosmotic to their surroundings. As a result, their kidneys will have many cortical nephrons. These nephrons will help to produce large amounts of dilute urine rapidly and also help to conserve salt, specifically in the distal tubules. Marine fish, on the other hand, have reduced nephrons, which typically have no distal tubule or a small or absent glomeruli. The kidney in marine fish functions to excrete divalent ions such as calcium, magnesium, and sulfate. They will excrete these ions into the proximal tubule to be eliminated. In addition, specialized chloride cells are present in the gills. These specialized cells will help to excrete excess salt, such as sodium chloride, to also aid in osmoregulation. We're now going to take a look at how the mammalian kidney is regulated. The volume and concentration of the urine produced can adjust to what the animal needs with respect to the animal's water and salt balance. As a result, the regulation of the kidney is closely related to both blood pressure as well as blood volume. There are three main pathways that are involved in regulating the mammalian kidney. Antidiuretic hormone, or ADH, is typically produced in the pituitary gland. This will activate receptors on the collecting duct cells to increase the number of aquaporins to maximize water resorption. The RAS system, or renin-angiotensin-aldosterone system, is released in response to dehydration. This causes an increase in water and sodium reabsorption, as well as an increase in blood volume and pressure. AMP, or atrial natriuretic peptide, is released by cells in the atrial walls to counteract the RAS. This will then cause an overall decrease in sodium chloride resorption, and it will lower blood volume as well as blood pressure. We're now going to take a look at each of these mechanisms. The first mechanism we will take a look at is the mechanism of antidiuretic hormone, which is also known as vasopressin. ADH, or vasopressin, is released from the pituitary gland in response to an increase in blood osmolarity above the normal range of 275 to 295 milliosmoles per liter. ADH will bind to specific receptors in collecting duct cells. This will start a signaling cascade that will increase the amount of aquaporins in the plasma membrane. Take a look at the diagram to the right to see the signaling cascade. When ADH binds to the specific ADH receptor on the surface of the collecting duct cell, this will cause the production of cyclic AMP, which is a secondary messenger. This will activate protein kinase A. This will lead to the overproduction of aquaporin, water channels and an increase in exocytosis, which will insert these aquaporins onto the plasma membrane. Because there are more aquaporins, more water can be resorbed. This will decrease urine volume and decrease the blood osmolarity. As the blood osmolarity will fall back within the normal range, ADH production decreases through a negative feedback loop. This diagram represents the overall mechanisms involved in the action of vasopressin or ADH. When blood osmolarity increases, this will trigger osmoreceptors in the hypothalamus. This will trigger the production and secretion of ADH from the posterior pituitary. When ADH is released, it will circulate in the bloodstream where it will then reach the kidney. ADH will bind to specific receptors on the collecting duct cells as seen in the previous slide. This will then increase the number of aquaporins in the membrane to increase the resorption of water. Also note that there is a behavioral response that occurs as well. When osmoreceptors in the hypothalamus are triggered, this will then lead to the generation of a thirst behavioral response. This will typically lead to an increase in water consumption. As a result, there is a twofold increase in water, through the consumption of water and through the resorption of water through the collecting duct. This will then restore the blood osmolarity back to the normal range. This would then initiate a negative feedback where the amount of ADH secreted by the posterior pituitary would then decrease. We're now going to take a look at RAS. RAS is the renin-angiotensin-aldosterone system for controlling the kidney. Sometimes blood volume and blood pressure may decrease without changing osmolarity. This can occur in periods of extreme blood loss or extreme diarrhea and dehydration. The RAS will cause an increase in water as well as sodium resorption in response. Specialized cells that are present in the juxtaglomerular apparatus of the Bowman's capsule will detect a drop in blood pressure or a drop in blood volume in the afferent arteriole. As a result, when this is detected, specialized cells will release renin enzymes. These enzymes will help to convert angiotensinogen to its active form, angiotensin. This will then cause vasoconstriction, reduce blood flow, and also stimulate the secretion of aldosterone from the adrenal glands. These are small glands that reside on top of each kidney. When aldosterone is released, it will increase water and sodium reabsorption. As a result, there will be an increase in blood pressure and volume. This would then inhibit the release of renin, shutting down this system. This diagram represents the mechanisms involved in RAS. When there is a drop in blood pressure or volume, sensors in the juxtaglomerular apparatus will detect this decrease. These specialized cells will then secrete renin. Renin will help to convert angiotensinogen into its active form, angiotensin, specifically angiotensin II. However, renin is involved in the conversion of angiotensinogen to angiotensin I. Angiotensin converting enzyme will then be activated to allow for the final active conversion to angiotensin II. Angiotensin II will act on the adrenal gland, which will then cause the release of aldosterone. Angiotensin II also leads to vasoconstriction. Aldosterone will help increase the reabsorption of water as well as sodium ions. As a result, this will help to restore the blood pressure and blood volume into the normal ranges, thus shutting down the release of renin as a negative feedback loop. We're now going to take a look at the last pathway, the AMP pathway, or atrial natriuretic peptide pathway. This pathway directly counteracts RAS to help decrease blood pressure and to also inhibit the release of renin. Specialized cells in the atrial walls of the heart will detect any increase in blood pressure or volume outside of the normal range. This will then cause the release of ANP. The release of ANP will inhibit the release of renin from the juxtaglomerular apparatus. It will also increase the excretion of sodium and water. This is because the sodium chloride reabsorption in the collecting ducts will be inhibited. This system also causes the inhibition of aldosterone release. As a result, sodium and water will be excreted and vasodilation will occur. This will cause an overall drop in blood pressure back to the normal range. The ADH, RAS, and AMP pathways work in coordination with each other to monitor and regulate kidney function in its ability to control blood volume, blood pressure, osmolarity values, and in maintaining a proper salt balance. In addition, the hypothalamus has neuronal circuits that will help in a behavioral response to control these aspects of the body as well. Activation of one neuronal set in the hypothalamus will stimulate an increase in thirst, while activation of a second set will halt the water consumption. Thus, an important behavioral as well as hormonal response all works to control the overall function of the mammalian kidney and the osmoregulation and excretion of materials.